Hello everyone, my name is Yeung. I'm from Burlingame High School, and I'm the co-founder of Iris with my partner Benjamin. And today we have the greatest pleasure to host Dr. Levitt right over here. Let's give him a round of applause. He's going to present his work that led to his Nobel Prize on molecular dynamics, and so that has really paved the way for many breakthroughs such as protein design and protein structure prediction, and even like nanomaterials. And so there were so many different applications that can come out of this. And yeah, it's very exciting work. I'm going to pass it off to my partner, Benjamin.
Yeah, thank you for that introduction, Lucas. So today we are very fortunate to hear directly from Michael Levitt and as high school students who are beginning to explore their own futures in science, technology, and beyond. This is an extremely unique opportunity to learn from someone whose curiosity and perseverance has changed the world. So please join me in welcoming Professor Michael Levitt.
Thank you so much. It's really great to be here. I hardly ever do talks like this, but just seeing how young you all look is really amazing. And when you get old, like a 50-year-old looks really young to me. And but it's great. I have grandchildren. It's sort of older than you and younger than you, even similar age. So anyway, you might actually ask, why do people who have won the Nobel Prize feel the need to talk to people? And I remember in California, you hear about the Nobel Prize at 2:30 in the morning. So unless you're crazy, you're asleep, and you wake up, and you got the phone and whatever, and then you suddenly have newspaper in the place, and CNN, and everyone is there, and the day's crazy, and the questions just keep on going. Not a few days later, what's all this fuss? I'm exactly the same person as I was 4 days ago. Nothing has changed. So why? And then you realize that when you were young, certainly in my case, there were people who really had a big influence on your life. And you suddenly realize that even if you didn't mean to get the Nobel Prize, once you have one, you sort of have the obligation to pass it forward. In other words, to try and tell young people that science is cool and science is fun. And for this reason, I actually am happy to do things like this, and I do quite a lot. This is the first time I'm doing it at Stanford, and I'm really grateful that this opportunity was given to me. Because really, this is going to sound quite obvious, but the future belongs to the young. And oftentimes, the old people think they're very important. Because for a Nobel Prize, like many prizes, you do the work when you're 25, and you get the prize when you're 60, 65. So basically, you kind of went, well, gee, I did the work 40 years ago, and only now they recognize it, but you were really smart when you were 25. Now you're kind of getting old. That's the way things work. People get recognition late because they want to make sure that it's really working and it's actually useful and so on. So it's important also to stress to young people that you guys are it. So I'm going to tell you a little bit about my own life in science. There'll be some science, there'll be a lot of personal history, and I called it A Wonderful Life in Science. So basically, I'm just going to start by saying a little bit about how modern biology began. I was born in 1947, baby boomer. But basically, in the years after the Second World War, huge steps were made in understanding biology. And I was 16, I was 13 years old in 1960, and at that time, I was already looking at Scientific American, a magazine you could buy in stores, and in there were lots of times really exciting articles about how life is really working. Also, whenever you succeed, you always succeed because you're standing on the shoulders of somebody who's come before. We're all where we are. We all have special environments. We all can use phones. We can use AI. All these things because people are there. And every generation should start out not at the bottom, not on the shoulders of the people, who went before. And Einstein. So, first person I want to mention is somebody called John Kendrew. He's a Nobel Prize. He was the first person to solve the structure of a protein. Protein is a really complicated thing. This is a picture of all the atoms in a protein. Even a small protein might have 2,000 atoms. And each of this, that's one atom, and that's another atom, and that's another atom. And all of these atoms are not just mixed up. They're all arranged in a very precise way in space. So Kendrew, in 1959, solved the structure of myoglobin. And getting proteins is quite hard. You have to do good biochemistry. And you have to get lots and lots of meat, if you like, to get the protein from the meat, because protein is usually meat. So he used myoglobin, which is a protein that's found in whales. Back in the 1960s, you could kill whales, and whales were being killed. And you get a lot of meat, so you can extract the protein. And myoglobin is a protein that is actually in the muscles of whales. And when whales dive into the ocean, they store oxygen in their muscles. Because if you're diving and you store them in your lungs get compressed. But the oxygen in your muscles is easy. So it's a protein that stores oxygen. The oxygen actually binds to this red object here. So he solved the first protein structure. The next person is somebody you probably all heard of, Francis Crick. He's the person who, together with James Watson, solved the structure of DNA. And he was actually working in the same laboratory as Kendrew. So basically, Kendrew solved the structure of the first protein. Crick and Watson told us about the structure of DNA. And we'll see how these connect. And then there's a person who maybe is even less well known, Roots. He also has a Nobel Prize. He and Kendrew and Crick and Watson all went to Stockholm at the same time. And Roots was actually working on a bigger protein than the myoglobin. It's a protein called hemoglobin. This is the protein that in our bloodstream carries the oxygen from our lungs to where it has to go to. And basically, each of those colored things, there's 4 here. 1, 2, 3, 4. Each of those is like a myoglobin. So this thing has 6,000 atoms. And each of these red things is a chemical group called a heme group, and 1 oxygen connected to this thing. So the machine is huge compared to oxygen. But biological machines are like that. Now, one interesting fact that hardly anyone knows is when Crick did his famous work on DNA, this is a paper that went into Nature, probably one of the most famous papers that were published, he was actually the PhD student of Perutz. But Perutz is not mentioned. No one felt the need to mention their PhD student because back then they felt that young people have to be given independence and responsibility. So things have changed a little bit in that way. Not in a good way. Okay. So these heroes of mine, and they really were my heroes. I read about them when I was 14 or 15, even younger, maybe 13, Scientific American, and you can see here, this is a page from Scientific American. Scientific American was really important in the 60s because it was the only journal that published in color. So its cover was in color. And all the famous scientific journals like Nature and Science and stuff, it's all black and white. And if you solve something as complicated as that, you'd like to see it in color. So basically, this is just a little bit about what is, if you like, the secret of biology. And in some ways, you could actually put all of biology onto one slide like this. The secret of life. Because basically, molecules are learning machines. And that's how they evolve. So basically, over here on the left, you have the DNA. And DNA is just like sentence. There's a sequence of letters and they spell something. What they generally spell is how you make a sequence of letters in a protein. I'll say more about this. The protein then just spontaneously folds up into a folded shape, and the shape is always the same. So in your body, you have a protein looking like this, and yours looks just like mine. And then the protein, because of its shape, maybe can bind food. There are different Chinese. Sorry. This talk is actually half Chinese, half English. And that's a food molecule, and then if that food molecule makes the cell live better, you make more of the DNA. So you learn by preserving what works. So DNA is a very pretty molecule. It consists of 2 chains, a red chain and a green chain. atoms. If you look at the atom level, it looks like it's the same everywhere. But actually it's not. And some places actually have 1 or 2 extra atoms that make the letters. So basically this thing has these 4 different kind of letters, and the letters are a code that's saying how you should make the proteins. Remember, nature worked this all out by itself. Scientists didn't do this. Nature spontaneously thought of this chemistry, and this chemistry is really smart. I mean, this is way beyond anything the greatest chemists can do. To think about how you store information in a molecule, be able to copy the information, and so on. Very, very smart. And then proteins, again, are like a long chain. But if you look at it carefully, the protein beads are different sizes. You can see even at the atomic level in the middle, oxygen is red and nitrogen is blue, and this is hydrogen and carbon, but some are big and some are small and so on. And they're essentially different colored beads. And this chain of different colored beads does something which is really remarkable. The chain, which is long, actually folds up into a cut shape. And that shape is precise and three-dimensional. So what a three-dimensional shape means is that that shape can interact with molecules. It can interact with other proteins. And the real world is shapes in 3 dimensions. Our world, when we pick up a hairbrush or a pen, they have different shapes, different functions. The proteins are like that, but the proteins are really, really tiny. This is probably magnified by 1 billion times, 1,000 million times. So this is the proof that in your body, they're so tiny, in fact, the width of this protein is about 2 nanometers. And in the very, very best chips on your iPhone, the thinnest wires are 5 nanometers. So in the space of the thinnest wire, you actually have an entire machine. So that is basically biology. So basically, my work was to basically take these shapes that you saw and try to bring them to life. How do they operate? How do they move? How do they do things? And this ended up being the very beginning of structural biology. And this is John Kendall again, the guy who solved the Nobel structure. So he did something really interesting. Got the Nobel Prize at the end of 1962. Usually very Stockholm in December, 10th of December 1962. Came back to England and immediately made a television series called The Thread of Life. And this television series was just on BBC television, only one channel in those days, so everyone watches it. And it was basically a course in the wonders of biology. And this is just the name of it. Now, this was shown in the winter of 1964. Now, my own background, I was actually born not in England, I was born in South Africa. And when I was 16, I had an aunt and uncle who lived in London. My mother said, why don't you spend your holidays visiting your aunt and uncle? I visited them. They were both scientists, very good scientists. Maybe my mother wanted to be a kind of scientist. What was more important for me was they had television. And I was totally addicted to television. I'd never seen television before because in 1962 there had been no television in South Africa. Britain got television quite early, probably a bit later than the United States, but many, many countries didn't have television in the 60s. And I started to watch television. And I watched everything. I remember it was the Winter Olympics at that time, things like this. But luckily, I actually watched Kendall's program. So then I decided not to go back to South Africa. I would finish school in England. I would then try to go to university in England. I did all that. And at the end of my university, which I did in London, I studied physics, I said I want to do a PhD with Kendall. So I wrote to Kendall saying, can I do a PhD? And the answer came up very quickly, we're not accepting any PhD students. But my life, I've been very lucky. And usually people who have succeeded are very lucky because they didn't have any problems at all. Luck is important. But I had friends who, although they were studying physics, wanted to go to business school. So they had this business mindset. So you know, in business, when people say they will do it, you come back with a counter offer. You don't say, okay, fine, you want to buy my car, you say, be cheap, or I'll give you 2 for the price of 1, something like that. So why don't you write back and say, if you can't come this year, can you come next year? That doesn't seem like a genius move, but actually, I don't think I ever heard anyone say it. In science, not business, I can and the student wants to come next year. So they say, oh, well, definitely come and talk to us and so on. So I went up there and I met these people I've been talking about, great and so on. And then a week or so later, I get a letter back from them saying, look, we really like you, but we cannot tell you today if we'll take you in a year's time. So you're going to skip a year, you're going to do nothing for a year, and then we'll see if we take you. And my friend said, are you crazy? What do you haven't learned anything. They still won't take you, you still don't know about your future. Because people were in a hurry in those days about their future. So anyway, I went back, I managed to bump into Max Kurtz and the cold guys, didn't have an appointment, I just went back, and I said, oh, I can't study for my final exams, I'm so worried about my future. He's come to my office and he said, look, I can't decide about you, because I'm running the lab together with John Kendrew, we'll decide about on Monday. And Monday everything changed, they decided to accept me a year later, as long as I did what they wanted me to do in between. What they wanted me to do is not what I wanted to do. They wanted me to go to Israel to work with the scientists there. So Israel, you know, a long way away, just been a war there, there's lots of wars in Israel. It didn't seem to me to be a place that I really wanted to go to. But they basically made it a condition. Either you go to Israel, we will accept you. And the reason they did this was because Kendrick had a connection with the scientific institution in Israel, called the Levitt Institute, and he knew that they were trying to understand what were the forces that explain how small molecules work. And he realized that a big molecule is like a bunch of small molecules. He has some of the structures, like globin was one solved by Kendrew, a few years later, lysine was solved. And in all of these things, you have things that look like chemical groups. For example, it's like a 6-membered ring. There's a 5-membered ring. There's a 6-membered ring. You know, there's CO groups. So basically, a protein is like a whole lot of small chemicals connected together. If the certain forces explain why small molecules have shapes, they should also explain why big molecules have shapes. And in Israel, he basically sent me to work with this person, who was actually much younger at the time, and so was I. And he was working with these PhD students and trying to understand the forces between small molecules, like I just said. And Maria Valshin was working with Listen, and also shared the Nobel Prize with me. And my job was to basically, I wrote the computer program that Valshin and Lipson were using for their work. Now, I didn't know how to write computer programs, so they gave me the manual to Fortran, and I guess in 3 months I could do the program. Kind of surprising, I'm not sure I could now. Maybe with AI I could, but I certainly couldn't myself. Anyway, people were smart when they're young. So basically, as part of this whole issue, is something called the potential energy. You all know that if I fall down the stairs, I'm going to bump my head because potential energy is released. So potential energy controls many, many things. And for molecules, you also can derive potential energy. So 2 atoms can't go through each other. But when they're immediately distance apart, they actually attract each other. If one atom is positively charged and one is also positively charged, they repel each other, and so on. So there's all these very simple things. This is not quantum mechanics, it's just classical mechanics. But you explain the system essentially as balls connected by springs. And that ended up being quite a powerful representation, and it allowed us to take the structure of a protein as determined by these crystallographers and see what happens if you minimize its energy. Does it break apart? Does it stay together? So that was one of my very early papers. And basically, the paper was just showing that you could take the energy, minimize it, and the structure generally improved, but didn't fall apart. So that was the first time that people had actually calculated the energy of a protein. The energy is very important because if you get the energy of a system, you can also tell how it's going to move. So if you think about a ball in a string, potential functioning, and once you know it, you know the spin backwards and forwards, and you can calculate what it's going to do. In exactly the same way, if you have the energy from the molecular system, you can tell how it's going to move. But before I get to that, I just want to mention a very early paper that I wrote in 1969. It's a sub-author paper in Nature, which is impossible today. But I'm the only author, and I think the paper was actually sent to Nature by Francis Crick, who I'm now working with. And he probably remembered how Bruce had sent his papers in Nature 20 years before. So life is interesting, right? He less than 20 years, 17 years. So basically, now I'm in England, I've done my PhD. I'm going to actually go back to Israel to work with Rosh Hashanah. And then we did the other parts of the Nobel Prize citation. Basically, it was for multi-scale modeling of complex chemical systems. So what is multi-scale? I actually didn't even know what it was until I got to Sweden. They told me in Swedish it's flerskalig, multi-scale. I didn't even think I was doing multi-scale modeling, but they liked the words. And I sort of realized afterwards, multi-scale modeling is sort of explained by something Einstein is said to have said. Everything should be made as simple as it can be, but not simplified. And that's what that's really saying is every problem is at the right level of simplicity. So imagine you're trying to work out who your best friend at school is and whatever. It's probably enough to think about people, the characters, and things like that. You don't have to worry about the atoms of the person. You don't really care that the person is moving quickly or moving slowly. It's personality. So you need to think about the right things for the problem. This is true in every problem. If I want to see how my hands move, it's enough to think about muscles and joints and things like that. I'm not worried about the hemoglobin molecules inside my blood. So you want to know what to exclude and what to think about. The real reason is that these systems are very complicated. If you keep all the detail, you can't do anything. On the other hand, if I said, okay, a human body is like a ball, it probably wouldn't be a really good model. Maybe a stick figure would be a really good model, but saying people like balls, you probably wouldn't capture enough of what's going on. So that's being too simple. So the simplest thing about a person is a stick. Probably wouldn't get you very far. We did two things in multi-scale modeling. Just saying every problem should have the right level of detail. So we did models which are coarse-grained models, where you kind of take the atoms and sort of forget about many of the atoms. Trying to get the general field. And we used this on proteins. Proteins are chains, so we kept the chain, but we left out many of the atoms. And this made it very possible in computers that were working back in 1974 to actually do calculations. Now, computers today are much, much, much faster than they were back then. Your phone is probably the biggest computer in the world in 1995. So it's kind of amazing how things have changed. We sort of forget about that. But we had to use linear computing, that's what we had. And basically, together with Arlo Varsho, we took a protein chain shown there, all the atoms, and just approximated it by just a few spheres. And then allowed the chain to fold up from its open state, slowly fold back to a complex. This is really the first simulation of a protein folding up. The answers were not very accurate, but it was the first time. This whole thing took maybe 20 minutes of computer time. We take like a microsecond on your iPhone. Okay. Then we did something in the other direction. We wanted to try to understand how enzymes carry out reactions. Enzymes are also catalysts. They don't just have shapes, but if a small molecule connects to the enzyme, they can break the small molecule into two pieces and then use the energy like food. So in our body, we're continually breaking molecules apart for food, energy. We're basically putting them together in some places, they're moving in other places to need energy and breaking them down. And basically, we had to make the protein more complicated so that we could do quantum mechanical calculations. And essentially, the idea was to put in electrons and protons. So instead of an atom being a single sphere, there would be electrons inside it and protons. But that makes the calculation very expensive. So you would only take a small part of the system and make it in this way. And this enabled us to actually do calculations on the enzyme reactions that proteins have. Okay. Then, a little bit later, 1979, I was already looking at how proteins move, and actually made the first movie showing how protein moves. Now again, it's hard for people to understand how difficult it is to make this back then. So here's a protein moving, and each of the spheres are moving. The protein is in a vacuum, so you don't see anything around it. This movie probably cost $500,000 to make. I didn't pay for it. Because back in those days, drawing something like this was really expensive. And the guy who decided knew how to shade the sphere was an employee of the NIH who went on to found Pixar movies, called George Porter. So it's just amazing how things were. And in this movie, you'll see a water molecule come drifting out, and it gives you a feeling of how the protein's moving. It's when I first saw this, I couldn't believe it. I think I saw the first 20 seconds, and now a couple of minutes. This movie was really the first movie made, the first use of computer graphics. If you think what you can do now, it's just kind of amazing. Okay. So if you look at the way the protein is moving, I mean, that didn't look. I keep on asking myself, what did it actually look like? And I kind of sometimes think, sometimes you get a bag of worms, they're all moving, they're not going anywhere. The reason they're not going anywhere is they're all tied together, except for that water molecule that wasn't tied to the rest of the structure that came out. So things are moving. They're not moving like a tree in a nice swaying way. Yeah? You're moving, jiggling around. And that I think is a good description of the molecule. So then we did some protein molecular dynamics another 10 years later, in 1988, in water. This is actually much more difficult because you have many more atoms in the water than you have in the protein. You can see a small part of the protein in water at low temperature, 278 Kelvin is 5 degrees centigrade, 45 degrees, 43 degrees Fahrenheit, quite cold. And it's basically not falling apart. The molecule is very happy. But now when you do this at high temperature, it comes apart. And we know that proteins come apart at high temperature. When you boil an egg, boiled egg is protein that's come apart. So now you're going to see the thing at high temperature, and it very quickly comes apart. This is much too high. I didn't have to make it so high. I made the movie, and all movies have a certain value, you have to redo it, but it's something like that. Let's give you a glimpse of how the protein actually moves when it's coming apart. Now more's happening. It's not just jiggling around, it's moving. Some of the movement is actually quite interesting. We'll see in a second. Back to space filling. And this is something you really couldn't observe experimentally. With only a calculation can reveal something like this. I think there's some more, yeah. So there you can see it kind of comes apart, a bit like a snake. The chain is thick and self-avoiding, and so on. Okay. So basically, what about the future? This is maybe the future as it was 10 years ago. We would like to simulate the motion of big machines, big protein machines. And the most exciting machines is something called the ribosome. It's a big machine. It has something like 200,000 atoms. We would like to see how this machine moves as it functions. And this machine is pretty amazing because this machine is the inkjet printer of life. It takes the message from the DNA and prints out proteins. So it takes one set of code and converts it to a different protein. But once you have made the proteins, they flip up. So it's kind of very straightforward. So basically, this is just a video of a cartoon of how a protein works. The DNA is the long message over there, and it reads 3 letters at a time, and tries to make the chain at the top that's growing. That's going to fit in with an adapter molecule or transfer RNA, which might release anti-cables about. That thing that looks like a thing now it's going to suddenly come across. I guess the reaction will go, yeah. And now it moves to the next one. But what does this actually look like for the real structure? So here's the actual motion in the real structure where the orange thing is the DNA or RNA. The adapters are those big green things, green and red things. And at the very end of the red thing is the amino acid that's going to give to the red one. This is a machine that operates for all proteins are made this way. And we have these in our body. These are the machines that are taking the instructions from the DNA and making proteins, which then fold up to do all the work. So it's kind of amazing chemistry. It's like you did in the system with build a house by just tying together the pieces and say, shoot, and the house will just assemble. I mean, nature is really, really smart. Okay, so this is close to the end. Just some general thoughts. So basically, I said there's been a big change in computers. And this slide is out of date. I actually made this slide in 2007, just after I got the Nobel Prize. Some newspaper called me up and said, how much faster have computers become? And you know, by this time, we were so sick and tired of saying the same nonsense to everybody and so on. Chance to think, let me really think about how a computer that I used back in '67, how it was now in 2007, 4 years later. What I found, rather to my surprise, that many things had changed. So the cost had gone down from $40 million to $4,000. This is my current laptop. It went down by 10,000 times. The speed had gone from 21 megahertz to a gigahertz had gone up 10,000 times. The memory had gone up 10,000 times, and the size had gone down 10,000 times. And I actually used this during my Nobel speech. Because basically I said, if cars were like computers, then a new Volvo 3 would cost $3, would have a top speed of 2 million kilometers an hour, would carry 50,000 adults in the park in a shoe. Now, that's funny, but it's actually true for computers. I mean, all of those things, size, memory, speed, capacity, have all increased so much. And all of those things matter. And this is already 2007. If I did it now, I'm sure you'd add a zero to every one of those, maybe even more. I mean, it's really incredible just how much computer power is involved. It's amazing. Okay, I think that is the end. So, my advice for you. So, be passionate. Do what you like to. You know, if your parents really want you to become a doctor, you don't like being a doctor, that isn't necessarily a good idea. Because let's imagine in medical school, you're actually very successful, and you pass all the grades, you become a doctor. All the time you would have much rather done something else. So maybe you want to get a grade. And basically, there are so many ways in life that you can do interesting things. Be persistent. You know, when Cambridge said to me, no, we won't take you, I didn't say, oh, okay, I'll do something different. In other words, the reason you have to be persistent is you have to believe in yourself. If you don't think you're good, you'll never convince somebody else you're good. So it's really important to believe in yourself. Be creative. What I really mean is be original. Every one of us is different. Even identical twins are exactly the same. And you have ideas that's really unique to you. So do that. And my final point is be kind and good. Now, this is part of my Nobel speech. So why should I be kind and good? And I said, you know, if you're nasty to everybody, you're pushing things, there's a lot more people that are not you than are you. If I'm rude to all of you, at some point, one of you is going to get a chance to get me back. It's just not worth it. And life is much, much nicer when you're kind of good. Now, my talk formally ends with one thing I do want to say. In the last 31 months, I have become completely crazy about AI. I don't do anything. This talk was written before. It does everything for me. I see a picture, I take a picture. What's AI about? I'm doing AI for everything, not because I don't know the answer. Sometimes I do know the answer, but AI is a very complicated thing. Maybe the next complicated thing to an AI is your smartphone. And even if you've been using the phone for 10 years, you can meet someone and say, did you know about this? I didn't know. Gee, I can do that. It's a very complicated area where there's always new things to learn. So I really want to understand how to best use AI. And I just don't use one. I use ChatGPT, I use Claude, I use Gemini. And I don't know what the school says about AI. So in your private time, play with AI. Because now what everyone is saying is, you won't lose your job to AI, you'll lose your job to somebody who plays with AI. Because basically, and what's also interesting about AI is that the way any uses AI is different from the way I use AI. Each person uses it in a very unique way. And that's amazing. I mean, it's such a complicated thing. You know, for example, I use it for reading papers, but I've never used it to write a paper. You know, I like it to read a paper, then I ask questions. Did they really do that? Are you sure? I don't believe it. You know, and it'll tell you, it's a please show me. The other thing also, one very, very important thing that scientists have. Scientists don't care if you tell them lies. Because scientists are always doing experiments or calculations, and most of the time the calculations are wrong. Either bugs in the program, you're doing experiments, you didn't get the conditions right, so you realize that things are generally wrong. As a result, you don't believe anything until you show that it's right. So basically, in some ways, science, you know, in the criminal justice system, you are not guilty until proved guilty. In science, you were wrong until proved right. The opposite. And that's a very important thing to know. You know, if you were to say to me, what are the most important things to know in the current world? One is to realize that a lot of what you see is wrong. Not in a bad way. I mean, as a scientist, you know, students tell you things that are wrong. I tell myself things that are wrong. I mean, there's a very fine line between things that are wrong and new ideas, because every new idea is probably wrong in some way. The other thing is, the other thing is just play with AI in a way that will improve you. I think we're living in an incredibly exciting age. I didn't believe I would ever live to see AI as powerful as it is. But you know, be yourself. And when somebody says this is the best way you can use AI, you probably shouldn't even look at that because they've already said your best way. And try it for all sorts of things. I mean, you know, for personal advice, my girlfriend doesn't seem to like me anymore. Incredibly good. And the reason they're so good is if you ask your brother, or your sister, or your mother, or your father, they have a bias. And maybe your mother doesn't like the boyfriend, or she loves the boyfriend. You get an answer that depends on that person. AI will give you an answer that is unbiased. And that I think is really important. Okay, so thank you all very much.
Okay, so we're going to dedicate the next chunk of time to anyone who has questions. So if you have a question, just come on over here and grab one.
We'll just speak.
Yeah. So in your research, you talked about how a main limitation was technology because it wasn't as revolutionized as it is today. What were some other main limitations that you encountered during your research that really stand out to you?
So, you know, the main limitation of research is not knowing what to do. I mean, the limitations of, I'm not sure if I had had much more powerful computing, I would have done the same thing. You know, very often you arrive at some kind of truth in a very strange way. It's not like, oh, I'm going to go straight there. You keep like an ant searching for food when there isn't much food. Most of the time, you're going down dead ends. But even now with AI, I will do stuff with it and it'll give me the answer, but it's really a wrong answer. But by thinking about its wrong answer, I realize what the right answer is. So very often, even, you know, it's very strange how you discover things. So I think, you know, I tell people, 78 years old, I tell them I'm actually 7 years old. Maybe 7 to 8. Because basically, I think 7-year-olds ask questions. And I think in the future, we need to be able to distinguish right from wrong. And we need to learn to ask questions. And I don't think there's anything special. It's not prompt engineering. It's like, you know, hey GPT, I don't know how to talk to you. What should I do? And that's a perfectly good question. Or hey GPT, I'd like help with my dressing. What would you suggest? You might say, take a picture of yourself and I'll tell you what to do. But he'll tell you. And if the answer is stupid, it's like, couldn't you give me a better answer? So it's like, you know, I tell people that AI, and I hope no one is going to be offended, is like a super smart, really gifted 12-year-old boy. So basically, he's not that mature, doesn't fully understand how everything hangs together, but is super smart. But it's up to you to use it in a clever way to get the most out of it. So asking questions is very much like, how you think about how you talk to a friend. Friends are all different. How do you get the most from them? And AI agents are like friends, or enemies, or whatever, but you have to push them along to get stuff from them. Okay. Where's the microphone? Just ask. I can't keep track of who's asking questions, so I don't know. Since you came around.
I just want to know, at what age did you know that you wanted to pursue science? Did you have any other passions? How difficult of a decision was it?
You know, I grew up in South Africa. I would say my main interest as a young teenager was parties and girls and maybe pool. But that actually ended up working in my favor because I had a friend, and my friends in school were always the worst ones. So they were much more fun. And I was okay, I was like in the top 5, but I was never the best. It was too short. But when I was 15, yeah, I had a friend who I used to play. It wasn't pool, it was snooker, but it's like pool. And I used to play pool. And places where you were pool bored, usually like bars and places like that. And I once came back from playing pool at 3 a.m. My mother was quite sure I had been murdered. She was really angry with me. She wanted to ground me. She said, instead of grounding you, I think you were already bored with school. Why don't you just try to skip school? And she paid for private teachers and whatever, and I managed to go to university very young. So that actually was very important. But becoming a scientist, I think it might have been when I saw, I was interested in physics. And when I saw that life was actually physics from that TV program that Ken showed, I think that did it. So I decided to stay in England, not go back to South Africa, I decided to, but it's unclear. I think often in life you don't realize things like, for example, I told you that it was really important that Stanford set history. It was all the Nobel Prize winning work was done there before I was 25 years old. But I didn't even want to go. Sometimes in life suggests things to you. I think it's actually quite good in life to not know exactly what you want to do. I think sometimes people have like my 5-year plan is, or my 3-year plan is, whatever. Oftentimes strange things happen, and being able to use them is really important.
Okay, so I remember you said that at a young age you chose like the path of biology, but were there any other passions you had at that time that possibly could have been another path you took?
So I remember when we came to intake and I thought to high energy physics. I kind of liked it. So I really liked physics and math. I didn't like biology, it's so messy. But suddenly I saw that biology is driven by physics. And physics is nice because it was basically mathematical principles. But I think it was just, I probably didn't really like computers because I remember when I was 13 or 14, hearing that a computer, many, could actually play tic-tac-toe. Crosses and zero. Trivial game, and it's a game where you can always win if you play the right way. But computers can do it. My God, computer can play a game. I know they can multiply and divide and subtract. They can play games, and that proves that, oh my God. So in fact, I was probably also interested in artificial intelligence, which actually did exist in the 60s, in the very beginning. But what was special about structural biology? They needed big computers to solve the structural proteins. And as a result, they had computers there. So maybe I just went after the computers.
You mentioned you got help from Francis Crick with your research, and you also mentioned that he was one of your idols. So I was just wondering how you were able to set up that opportunity and any advice you have to those who want to learn how to make those connections.
I think if you want to interact with people, the story about Francis Crick is a little bit accidental because he had met a very close friend of my wife. I remember she was people in their 20s he had been visiting in Israel. given him a book to bring us. And we had a meeting for this reason. But things like that happen. We went and it was fun, and I guess, I think you can put yourself up. I think life is like lots of things are chance. And it's like a lottery. You think, okay, if it's a lottery, I don't have a chance. In a lottery, you have to buy tickets. And something happens, you try, put yourself out there. I think trying to be out of your comfort zone, which is really hard when you're young, it's much easier when you're old, is a really great idea. Try to expose yourself to interesting news. Just follow. There's so much cool stuff. And the other thing is, with the internet, it's amazing what you can learn. Stuff you can learn from TikTok and YouTube and stuff like this. It isn't all good. But it's always been the case. You choose. And you never know what's going to give you a good idea. There's some stupid performance. What? I mean, think about it. It's really interesting. So oftentimes, what triggers new ideas is it's usually not, I need a good idea. What's my first good idea? No, you don't do that. You just kind of go for a walk, take a shower, and get a new idea. It's because so I think these random things are very helpful.
I have a question. So, as you know, the diffusion model cracked.
Okay.
The diffusion model cracked protein design, and the transformer model cracked structure prediction. What do you think is like the next application of AI toward other biological discoveries?
I think there's a special circumstance. I don't know how much of science is going to be AI. I think a lot of science is going to be AI. But I still believe that the human AI combination, it's not like, it's really nice having a smartphone. And if somebody took your smartphone away from you, you feel pretty stupid until you got used to it. But the smartphone is not going to do anything by itself. Now, AI has more agency than a smartphone. But I think there's lots of strange things. People now are talking about vibe coding, where you just tell a computer program, write me a computer program to do this. Well, when I start writing computer programs for my science, and I use AI all the time, I don't even know what I want to do. If you say, well, please give me a website looking like her website, that's an easy question. And I'm sure the computer can do that very easily. But if you say, well, I'm trying to understand why COVID was so bad in the USA, for example, you don't even know where to start. And if you do, you'll say, you got this wrong, you have to fix it. So I use the computer for coding all the time. But they make lots of mistakes. So, I'm actually working with, sometimes I do the same problem, just read it as language models to write the code in different ways and compare it. But I don't think it's going to be quite so simple. And I think that, there's no doubt that could be amazing. Recently, I gave some handwriting that I couldn't interpret to GPT and got it right. It was my handwriting. I was amazed it could be my handwriting better than I could. And this is what I said, but it's surprising. On the other hand, sometimes I was going to do stuff and it's really stupid. So I think it's hard to predicting the future is basically not a good idea. Because you're very likely to be wrong. And I think there is no doubt that AI is going to be part of our future. I believe that it's the human-AI connection that's essential. Whether in some number of years humans will be unnecessary, I don't think so. We're relatively cheap. We don't use that much energy, and we do interesting stuff. But I think, one trouble with these AI things is that it's very hard to get a feel for. You really, and people tell you what they did, it doesn't make sense. Until you actually, I think I've asked AI maybe 20,000 questions in the last 21 months, starting with 3.5, and just keep on going, and ask them, I go to art gallery, I write contemporary, curator of contemporary art, and I just take pictures. And then I say, well, can you describe my trip? Why is it, it's amazing. I know stuff that even, back these, why did this painter paint this, and this one did this, very interesting stuff. Yeah, we're actually living together, we're competing, something like this. You find out things that are amazing just from a picture. So I just find that, but sometimes it doesn't do it right. saw a picture, an old picture, taken in New York, from some place in Central Park. And I asked 3 different AI, and I kind of guessed where it was, because it was a certain building at a certain angle, and I recognized the building. And I asked 3 different AI agents, and they all got it wrong. Because they didn't realize, because there's too many facts, and they knew it was Central Park, they knew it was New York, but they didn't get it right. So sometimes, we still have a lot to do, and I think what's going to happen, I mean, the stuff we used to worry about a lot before we had smartphones, like memorizing numbers, we don't care anymore. And we're fine. We just basically like standing on the shoulder of giants. Now we stand on the shoulder of technological giants. You want to stand on the shoulders of AI and be yourself.
I know you talked about your heroes growing up, and I was wondering what you thought about our current generation's scientific discoveries like gene editing.
Okay, so you were asking me about my heroes. Science has changed. And one way that science has changed, maybe I shouldn't be talking to you about this way, but I was writing an independent first author paper when I was 21 years old. And today it's almost unheard of. Because I was actually working at a PhD student, but I was in a research environment. Nowadays, I think the same baby boomers like me think that they're going to live forever and control everything forever, and they've really screwed up, not putting more faith in young people. Because the future is young people, and current scientists are not pushing forward young people enough. I give a lecture about this, and I tell them, look at the big scientific discoveries of the last 20 years. Where did they all come from? The things that really changed our lives. So, they all came from people who dropped out of Stanford University. And that's kind of true. I mean, this has had more effect on our world than 20 Nobel Prizes. Now, it might have been based on 20 Nobel Prizes. Or if you think about OpenAI, DeepMind, I mean, these companies are doing really incredible things. I was talking to some very important people in CS, and I said, aren't you ashamed that Transformers came from Google, not from Stanford? And they should be ashamed. And, so I think it's complicated. I think one of the great things of being a scientist is that, in principle, you can do things yourself. Now, maybe today I would say, oh my God, I'm doing my 3rd postdoc, and I still can't get a position, and people keep telling me what to do. But I was very lucky, I could do my own things. So maybe it's not the right way, I don't know. Something I think that universities are not going to laugh at. Because you can learn, you can go to AI and say, I'm really interested in learning biology, please make a course for me. You can do it. And then they'll ask you multiple choice questions, and very quickly. And you say, well, I'm really interested in learning biology, and I want it suitable for somebody who just tips up all day. And you'll probably get something amazing from just asking that. But very often when you predict where real discovery actually happens is when you take people who are different and say, do something together. So you pair together different people, you get amazing things. And you say, that this isn't happening enough. So I think this is something that science needs to encourage. One of the good things is that science is a global activity. The United States was not very good in science until 1945. Then became much, much better. Other countries are also around. So I think one of the good things about the world is that scientists have to retire everywhere else except in the USA. So young people actually have lots of opportunities in other places. It's unclear. I think the future is still incredible. And every one of you has an assistant. You wouldn't believe could exist 3 years ago. This is amazing. But I think you have to try to add be part of the science fiction. And there's an amazing science fiction book. One trouble with reading on Kindle is you never actually see the cover. I'm going to find it just because. everything in between. It's a book called The Diamond Age. It's actually a book by a historical fiction writer, science fiction writer, called Neil Stevenson. And this is about a girl who gets like a magical phone that can talk to her, can help her. And basically that's what we're in. This is, I mean, when I read the book 4 years ago, it seemed like complete nonsense. So you guys are living in a very, very special time. You have to realize that nobody knows what your future is going to be. But you have to be ready. Okay. What's going to happen next? Don't leave.
We're not leaving.
Okay, well, let's have one more round of applause to everyone. Okay, so everyone's staying here right now, but in a few minutes we're going to head over to the lab where the tour is going to be. And yeah, just put
As our sincerest gratitude, we have a gift for you, Dr. Levitt. Thank you so much. It's an absolute pleasure. I'm sure many of you guys are inspired. Many of you guys are inspired. And yeah, this is incredible work. And there's going to be even more inventions from him, other people. So yeah, let's give one more round of applause.